Hello and welcome back to the Sleepy Darwin. Whether you're here to drift peacefully into sleep, to unwind from the complexities of your day, or simply to let your mind wander through the ancient mysteries of life itself, you've found exactly the right place. Tonight, we're embarking on a journey that began nearly 1 billion years ago in primordial seas, when a single, unremarkable cell made a choice that would echo through deep time and eventually lead to you, lying there now, breathing softly in the darkness. This is the story of how one tiny decision to stay together, rather than drift apart, gave birth to every creature that has ever lived. From jellyfish pulsing through ancient waters to tigers stalking through tall grass, from hummingbirds hovering at flowers to humans contemplating their own origins, it all traces back to that first moment of cooperation. Before we begin this gentle voyage through evolutionary time, if you find these sleepy explorations soothing, a like and subscribe would mean more than you know to a creator whispering the secrets of life into the quiet night. And please, share in the comments where you're listening from and what time it is in your corner of the world. Are you curled up in a bustling city or nestled in countryside silence? Fair warning, the story of life is remarkably beautiful, so I hope this isn't so fascinating that it keeps you wide-eyed with wonder. But if it does, at least you'll drift off knowing how every cell in your body carries the memory of that first ancient partnership. Now settle in, let your breathing slow, and let's trace the impossible journey from one to many, from simple to complex, from that first lonely cell to the magnificent diversity of animal life that surrounds us today. Close your eyes for a moment, and let's travel together. Not through space, but through time. Deep, deep time. We're drifting backward through the centuries, past the rise and fall of civilizations, past the first humans painting on cave walls, past the dinosaurs with their thunderous footsteps, past the first forests whispering in ancient winds. We're going back almost 900 million years, to a time when our planet was almost unrecognizable. Imagine, if you will, an ocean unlike any you've ever seen or dreamed of. There are no fish darting playfully through coral reefs in rainbow flashes of silver and gold. No whales rising majestically to the surface, their songs echoing through the deep blue chambers of the sea. No dolphins dancing through the waves. No jellyfish pulsing like living umbrellas. No sea turtles gliding with ancient wisdom through underwater gardens. Instead, picture endless blue-green water stretching to every horizon. Water that shimmers not with the movement of schools of fish, but with something far more subtle, far more mysterious. The ocean is alive, yes, but with a different kind of life entirely. It's teeming with countless billions of tiny, invisible architects of the future single cells, each one a complete universe unto itself, drifting through the vast liquid cosmos of early Earth. The water itself has a different quality than the oceans you know. It's rich with minerals and nutrients, like a primordial soup that's been simmering for eons. The atmosphere above holds less oxygen than you're used to breathing, and the sun, filtering down through the water, creates patterns of light that dance and shift in ways that seem almost expectant, as if the very ocean is waiting for something extraordinary to happen. There are no beaches as we know them, no familiar shorelines where waves lap gently against sand. The continents themselves are different, clustered together in arrangements that would look alien to our modern eyes. And in this strange, beautiful ancient world, something remarkable is about to unfold. Something that will, in time, lead to every creature that has ever lived, including you, lying there now, breathing softly as you listen to this story. The silence of this ancient ocean is profound. There are no whale songs, no splash of jumping fish, no cry of seabirds overhead, just the gentle whisper of water moving with the tides, and the soft, nearly inaudible symphony of billions upon billions of microscopic lives, each one carrying within it the seeds of all future possibilities. And somewhere in this vast, quiet ocean, let's imagine we can focus our attention on one particular cell. She, and yes, let's call her she, because every great story needs a protagonist, and she is about to become the most important character in the history of life on Earth. She was unremarkable in many ways. A tiny blob of life, barely visible even if there had been eyes to see her, which there weren't. Not yet. If you could shrink yourself down and swim alongside her, you might think she was almost ordinary. She was roughly spherical, about the width of a hair on your head, enclosed in a delicate membrane that separated her inner world from the vast ocean around her. This membrane was like the gentlest of boundaries, porous and flexible, allowing nutrients to flow in and waste to flow out, but maintaining the essential integrity of her being. Inside her membrane, her world was surprisingly complex and beautiful. She was what scientists would eventually call a eukaryotic cell, a cell with a nucleus, that precious inner chamber where she kept her genetic treasures. Think of her nucleus as a library, containing all the information needed to build and maintain life, written in the elegant language of DNA. This library held the instructions for making proteins, those molecular machines that would carry out the work of living, and most importantly, it held the blueprint for making more cells exactly like herself. Floating around her nucleus, like tiny islands in her inner sea, were organelles, specialized structures that each had their own jobs to do. Her mitochondria, those remarkable powerhouses that you still carry in every cell of your body, were busy converting nutrients into energy. These ancient engines hummed with quiet efficiency, breaking down simple molecules and releasing the energy that powered all of her life processes. They were once free-living bacteria themselves, ancient immigrants who had joined forces with her ancestors in one of life's first great partnerships. She had other organelles too, structures for building proteins, for storing materials, for maintaining the delicate chemistry that kept her alive. Her cytoplasm, the gel-like substance that filled her interior, was constantly in motion, streaming and flowing like a microscopic river system, carrying materials where they needed to go. But perhaps most remarkably, she carried within her the capacity for something that would seem almost magical. She could make a perfect copy of herself. Every piece of information needed to build another cell, exactly like her, was contained in those coiled strands of DNA in her nucleus. She was, in essence, both the blueprint and the builder, both the recipe and the chef. From the outside, as she drifted through the ancient waters, she might have seemed peaceful, almost lazy. But inside, she was a bustling metropolis of molecular activity. Thousands of chemical reactions were taking place every second, each one precisely coordinated with all the others. She was constantly sensing her environment, adjusting her internal chemistry, making the countless tiny decisions that kept her alive in a world that was often harsh and unforgiving. She had no eyes to see the beauty of the sunset filtering through the water above her, no consciousness as we understand it to contemplate her place in the universe. And yet, in her own way, she was already perfect, a masterpiece of biological engineering, refined over millions of years of evolution into something that could survive, thrive, and most importantly, reproduce in the ancient seas. Day after day, in a rhythm as old as life itself, she would drift and divide, a cosmic game of solitaire, played with the building blocks of life. Her existence followed patterns that were both simple and profound, driven by the basic imperative that has motivated all life since the very beginning: survive and pass on the instructions for life to the next generation. When nutrients were abundant, and in those ancient, mineral-rich waters, they often were, she would grow. Slowly, methodically, she would absorb the simple molecules around her: carbon compounds, nitrogen, phosphorus, sulfur, and trace metals. Each molecule that crossed her membrane became part of her story, incorporated into the complex molecular machinery that kept her functioning. The process of growth was like watching a master craftsperson at work, though it unfolded over hours rather than minutes. Her cellular machinery would take those simple building blocks and transform them into the more complex molecules she needed. Proteins that acted as tools and structural components, lipids that maintained her membranes, carbohydrates that stored energy for lean times ahead. As she grew larger, something wonderful and inevitable would begin to happen. Her DNA, those precious genetic instructions, would start to copy themselves. This process was carried out with extraordinary precision, like a master calligrapher reproducing an important document. Each chemical letter had to be matched with its proper partner, each gene duplicated exactly, so that the copy would contain all the same information as the original. And then, when she had roughly doubled in size and successfully copied all of her genetic material, the most remarkable moment of her life would arrive: division. Slowly, carefully, she would begin to pull herself apart. Her membrane would start to pinch inward, like a balloon being squeezed in the middle. Her copied DNA would separate, with one complete set moving toward each end of her elongating body. The moment of actual division was both dramatic and gentle. One moment there was one cell, and the next moment there were two, each one carrying the complete blueprint for life, each one ready to begin the cycle anew. It was a kind of magic that happened billions of times every day in that ancient ocean, the magic of life, creating more life. But here's what made her existence both beautiful and haunting. Each time she divided, she ceased to exist as an individual. The she we've been following would split into two new individuals, each carrying her memories encoded in DNA, but neither one being exactly the same entity that had existed moments before. In a sense, she achieved immortality through this process. Her genetic information could potentially live forever, but she paid for it with her individual identity. Imagine living a life where your greatest achievement, your moment of ultimate success, was also the moment of your disappearance as a unique being. Every time she reproduced, she was simultaneously creating the future and ending her own personal story. And so her daughters would drift away from each other in the gentle currents, each one beginning her own cycle of growth and division. There was no family reunion, no passing down of wisdom from mother to daughter. Each cell was born with all the knowledge she would ever need already encoded in her genes, and each was destined to live the same solitary existence as her mother before her. The loneliness of this existence is almost impossible for us to comprehend. She had no companion to share experiences with, no mate to cooperate with in the business of living, no offspring to nurture and protect. She was complete unto herself, but also completely alone. Her interactions with other cells, when they happened at all, were brief and impersonal. Perhaps a chance encounter as currents brought them close together, or competition for the same nutrients in a particularly rich patch of water. Yet there was a kind of meditative peace in this solitary existence. She needed no one, depended on no one, competed with no one, except in the most abstract sense. She was like a tiny monk drifting through the oceanic vastness, focused entirely on the simple practices that sustained life: grow, copy, divide, repeat. Her life was pure in its simplicity, elegant in its self-sufficiency. But something was stirring in the ancient waters, a whisper of possibility that would change everything. The ocean that had been her stable, predictable home for countless generations was beginning to shift in subtle ways. Perhaps it was the slowly changing chemistry of the water itself, as other microorganisms altered the balance of nutrients and minerals. Perhaps it was the gradual cooling of the planet, or changes in the patterns of ocean currents that brought new challenges and opportunities. Or perhaps, and this is one of the most beautiful possibilities, the change was coming from within life itself. After millions of years of perfecting the art of solitary existence, some cells were beginning to experiment with something revolutionary, staying close to each other after division, instead of drifting apart into isolation. Picture this. Somewhere in the vast blue expanse, a cell very much like our protagonist was going through her familiar ritual of division. But this time, something was different. Maybe it was a small mutation in the genes that controlled her membrane, or perhaps a change in the proteins that normally helped daughter cells separate cleanly. Whatever the cause, when division was complete, instead of the two new cells drifting apart as they always had, they remained together. At first, this might have seemed like a mistake, even a disability. After all, staying together meant sharing resources, competing with each other for nutrients in the immediate vicinity. It meant that when one cell moved, the other had to move with her, limiting their individual freedom to explore the ocean. But slowly, almost imperceptibly, some of these accidentally connected cells began to discover something remarkable. Together, they could do things that neither could accomplish alone. Two cells attached to each other could sense their environment more effectively, with twice as many molecular sensors examining the water around them. They could process nutrients more efficiently, with one cell focusing on one type of food while her sister specialized in another. Most intriguingly, they found they could communicate with each other through chemical signals, molecular messages passed through their shared connection. For the first time in the history of life on Earth, one organism could essentially talk to another, sharing information about the world around them. The first stirrings of this change must have been subtle, almost invisible against the backdrop of the vast ocean. Here and there, pairs of cells would remain connected after division, forming tiny partnerships that lasted for hours, days, or even weeks before eventually separating or dying. Most of these early experiments in togetherness probably failed. The coordination required for two independent organisms to live as one was extraordinarily challenging. But evolution is patient, and evolution is creative. Every accidental success, every brief moment when cooperation proved more beneficial than independence, was like a vote in favor of this new way of living. The cells that could stay together successfully had advantages, and advantages in the ancient ocean meant better survival, more opportunities to reproduce, and more chances to pass on the genetic changes that had made cooperation possible. Meanwhile, environmental pressures were mounting. The ocean was becoming more crowded as life diversified and multiplied. Competition for resources was intensifying. Predation, larger cells engulfing smaller ones, was becoming more common and more sophisticated. In this changing world, the old strategy of solitary existence was becoming less reliable, less safe. It was in this context of increasing challenge and opportunity that our protagonist found herself living. She was still fundamentally the same type of cell that had thrived in the peaceful waters of earlier eons, but she was also poised on the edge of the most important transition in the history of animal life. Can you imagine being the only you in existence, the only individual of your kind? Think about it for a moment as you lie there, surrounded by the warmth and comfort of your own life. You are connected to other people through countless invisible threads: family, friends, community, the whole vast network of human relationships that gives your existence meaning and context. But our ancient protagonist knew none of this. She was utterly unique and utterly alone, carrying within her genetic code the entire future of animal life, but having no way to know the momentous role she was about to play in the greatest story ever told. Every division she underwent could be the one that led to the first successful partnership. Every daughter cell she produced could be the one that would choose cooperation over isolation, laying the foundation for everything from coral reefs to human civilizations. The future hung in the balance with each gentle pulse of her cellular machinery, each careful copying of her DNA, each moment when she chose to divide rather than remain as she was. And perhaps most remarkably, this choice, when it finally came, would not be made with any awareness of its consequences. She would not know that she was about to invent the concept of us in a universe that had only ever known I. She would not realize that her simple decision to stay connected to her daughter would echo through deep time, eventually leading to creatures that could contemplate their own origins, that could lie in bed on quiet evenings and marvel at the ancient journey that had brought them into existence. The stage was set, the players were in position, and the greatest drama in the history of life was about to begin. In the vast, ancient ocean, countless billions of solitary cells continued their eternal dance of growth and division, unaware that one of them was about to change the rules of existence forever. The lonely cell drifted through the blue-green waters, carrying within her tiny form the seeds of every heartbeat that would ever beat, every eye that would ever see, every mind that would ever wonder about its place in the cosmos. And in the gentle currents of that primordial sea, change was stirring like the first whisper of a wind that would eventually become a hurricane of evolutionary creativity. The age of solitude was drawing to a close. The age of cooperation was about to begin. And then, one day, though day hardly seems the right word for such a momentous shift, when time itself seemed to move with the patient rhythm of geological ages, something extraordinary happened in the vast blue theatre of our ancient ocean. Picture, if you will, a moment that would make even the most spectacular fireworks display seem pale in comparison, though to any observer floating in those primordial waters, it would have appeared utterly unremarkable. Our protagonist, that tiny architect of the future we came to know in the previous chapter, was going through what seemed to be just another ordinary cycle of growth and division. She had been drifting through a particularly rich patch of ocean, where mineral-laden waters had created a feast of nutrients. Over the course of several hours, she had grown plump and healthy, her cellular machinery humming with efficient productivity. Her DNA had been carefully copied, each strand duplicated with the precision of a master scribe reproducing a sacred text. The time had come, as it had countless times before, for division. But this time, and perhaps it was the unique chemistry of this particular patch of water, or maybe a cosmic ray that had traveled billions of miles to strike her DNA at just the right moment, or simply the accumulation of tiny changes that had been building over generations, this time was different. As she began the familiar process of pulling herself apart, something new stirred in her cellular machinery. Deep within her membrane, proteins that had never quite worked together before suddenly found themselves in perfect alignment. Chemical signals that had always been present but dormant suddenly awakened, like sleepers stirring at dawn. The division proceeded normally at first. Her membrane began to pinch inward, creating that characteristic figure-8 shape as she prepared to become two. Her duplicated DNA separated obediently, one complete set moving toward each pole of her elongating form. But then, just as the final separation should have occurred, something unprecedented happened. Instead of the clean break that had characterized every division in her lineage for millions of years, she hesitated. Her membrane, rather than sealing completely at the point of separation, maintained a delicate bridge between what should have been two independent cells. It was as if some part of her, some deep, molecular intuition, recognized that this moment was too important for a complete goodbye. And so, for the first time in the history of complex life on Earth, one became two, and stayed. The daughter cells, each carrying identical copies of their mother's genetic legacy, found themselves still connected by the thinnest thread of shared membrane, still able to exchange molecules and chemical signals, still fundamentally united despite their physical division. From a distance, they might have looked like two soap bubbles that had bumped into each other and stuck, their shared boundary creating a gentle curve where their individual spheres met. But this was no accident of surface tension. This was evolution taking its first tentative step toward one of the most profound innovations in the history of life, true multicellularity. The moment stretched on, and the connection held. Where every previous division had resulted in immediate independence, these two cells remained together, drifting through the ancient waters as a pair, their movements coordinated by their physical connection, their biochemistry beginning to synchronize in ways that neither could have achieved alone. It was, in every sense, the birth of us, in a universe that had only ever known I. And like many births, it was both miraculous and utterly natural, the inevitable result of countless small changes building toward a moment of transformation that would echo through deep time. Now, you might be wondering, and it's such a delightful question to ponder as you rest there in your own comfortable space, what exactly kept these pioneering cells together when all their ancestors had drifted apart? The answer lies in one of the most elegant solutions life has ever devised, a molecular innovation so important that you carry refined versions of it in every cell of your body right now. Think of it like cosmic Velcro, invented by life itself. Just as Velcro works through thousands of tiny hooks grabbing onto loops, creating a strong bond from many weak connections, cells developed their own sophisticated attachment systems through proteins that could reach across the gap between membranes and hold on tight. These cell adhesion proteins, and what beautiful, almost poetic names scientists would eventually give them. Cadarins, integrins. Selectins were like molecular hands reaching out from one cell to grasp the hands of its neighbor. Each individual connection was relatively weak, easily broken by the right circumstances, but together they could create bonds strong enough to keep cells united, while still allowing the flexibility needed for growth and movement. The cadherin proteins, for instance, worked like tiny magnets embedded in the cell membrane. When two cells came close enough, these proteins would recognize their counterparts on the neighboring cell and lock together in a molecular handshake. But unlike a simple magnet, this handshake was dynamic, capable of strengthening or weakening based on the signals the cells were sending to each other. Inside each cell, these adhesion proteins were connected to an intricate network of structural elements. Imagine a microscopic skeleton made of protein fibers that could push and pull, contract and expand, giving the cell shape and allowing it to exert forces on its neighbors. When cells pulled on their adhesion connections, they weren't just holding hands, they were actively communicating, sending mechanical signals that could influence their partner's behavior, growth, and even gene expression. The chemistry of staying together was even more sophisticated than the mechanics. Cells began to develop elaborate communication systems, releasing molecular messages into the tiny spaces between them. These chemical signals were like whispered conversations, allowing one cell to tell its neighbor about the availability of nutrients, the presence of threats, or changes in environmental conditions. Some of these signals were simple, small molecules that could diffuse quickly from one cell to another, carrying basic information about metabolic state or stress levels. Others were more complex protein messengers that could bind to specific receptors on neighboring cells, triggering cascades of internal changes that would coordinate the behavior of the entire small colony. Perhaps most remarkably, these early cellular partnerships began to develop something like a shared identity. Through constant chemical communication and mechanical coordination, the individual cells started to behave less like separate organisms and more like parts of a larger whole. Their internal clocks began to synchronize, their growth patterns became coordinated, and their responses to environmental changes became unified. The forces that held these first cellular partnerships together were both gentle and strong, like the surface tension that shapes a dewdrop or the gravitational pull that keeps planets in orbit. They represented a fundamental shift in how life organized itself from pure competition between individuals to cooperation within groups, from isolation to community, from the singular to the plural. These first partnerships were tentative, like shy dancers learning to move together after a lifetime of solo performances. Imagine the delicate negotiations that must have taken place at the molecular level, as cells that had evolved for millions of years to be completely self-sufficient suddenly found themselves dependent on cooperation with another organism. The earliest successful colonies were probably quite small, just 2 cells, or perhaps 4 or 8, clustered together in loose associations that looked almost accidental. They moved through the water with a different rhythm than their solitary ancestors, their swimming patterns influenced by the need to coordinate with their partners. Where a single cell could dart and turn with individual purpose, these small colonies had to negotiate every movement, creating flowing, synchronized motions that were beautiful in their own right. Picture them drifting through the ancient seas like tiny living rafts, each colony an experiment in cooperation. Some partnerships lasted only hours or days before the stresses of coordination proved too great and the cells separated. Others found harmony quickly, their chemical conversations flowing smoothly, their mechanical connections perfectly balanced between holding tight and allowing freedom of movement. The successful colonies began to develop what we might call personalities, distinct patterns of behavior that emerged from the interaction between their component cells. Some colonies were cautious, clustering tightly together and moving slowly through the water, prioritizing safety and stability. Others were more adventurous, maintaining looser connections that allowed for rapid exploration of new territories. As these colonial experiments continued over thousands of generations, something wonderful began to emerge: diversity of form and function within the colonies themselves. Not all cells in a colony needed to be identical anymore. Some could specialize in swimming, developing stronger flagella and more efficient propulsion systems. Others could focus on feeding, optimizing their nutrient uptake mechanisms. Still, others could become sentinels, specializing in detecting and responding to threats or opportunities in the environment. We can see echoes of these ancient innovations in organisms that still exist today. Take Volvox, that beautiful sphere of cells that rolls through freshwater ponds like a living green marble. Each Volvox colony contains hundreds of cells, most of them focused on swimming and photosynthesis, but some specialized for reproduction. The colony moves as a unit, coordinating the beating of thousands of tiny flagella into a synchronized swimming motion that would make an Olympic synchronized swimming team envious. Or consider the chonoflagellates, those delicate collar-shaped cells that form temporary colonies as they filter nutrients from the water. When food is abundant, they live as individuals, but when resources become scarce, they come together in organized groups where cooperation increases everyone's chances of survival. They're like microscopic versions of pioneers circling their wagons against adversity, but infinitely more elegant. These modern organisms give us glimpses into what those first colonial experiments might have looked like. We can imagine colonies that formed beautiful geometric patterns, chains of cells linked like molecular pearls, or hollow spheres where each cell contributed to the structural integrity of the whole. Some probably looked like tiny flowers, with cells arranged in radial symmetry around a central point. Others might have resembled miniature trees, with branching patterns that maximized their surface area for nutrient absorption. The variety of experimental forms must have been extraordinary. Evolution was trying out every possible way that cells could stick together and benefit from the arrangement. Most of these experiments failed. Of course, cooperation is difficult, and the molecular machinery needed to make it work reliably took time to develop and refine. But the successful innovations spread through the ancient seas. each one representing a small victory for the power of working together. What's particularly beautiful about this phase of evolution is that it established patterns of cooperation that we still see today. The basic principles of cell adhesion, communication, and coordination that developed in those first simple colonies were refined and elaborated over hundreds of millions of years, but never fundamentally changed. The molecular handshakes that held together the first partnerships are still holding your body together right now, allowing your trillions of cells to function as a unified whole. Suddenly, our tiny pioneers discovered something remarkable. Together, they could do things that alone seemed impossible. It was as if they had discovered the secret of multiplication, not just in the mathematical sense, but in the sense that their combined capabilities were far greater than the sum of their individual parts. The first and most immediate benefit was protection. A solitary cell floating in the ancient ocean was vulnerable from every direction. Larger predatory cells could approach from above, below, or any side, engulfing their prey before the victim even knew danger was present. But cells in a colony could watch each other's backs, quite literally. With multiple sets of molecular sensors scanning the environment, threats could be detected earlier and from more directions. When danger did approach, colonial cells had options that solitary cells lacked. They could coordinate their escape responses, with some cells focusing on rapid swimming, while others maintained structural integrity. They could even engage in primitive forms of collective defense, releasing chemical deterrents more effectively as a group, or presenting a larger, more intimidating target to potential predators. But perhaps even more important than protection was the revolution in resource gathering that cooperation made possible. Imagine trying to eat a meal while also watching for danger, navigating through the water, maintaining your internal chemistry, and preparing for reproduction all at the same time. It's exhausting just to think about, and that was the reality for every solitary cell. Colonial cells could begin to divide these responsibilities. Some individuals could specialize in feeding, optimizing their molecular machinery for detecting and absorbing specific types of nutrients. Others could focus on locomotion, developing enhanced swimming capabilities that could carry the entire colony toward richer feeding grounds. Still, others could serve as scouts, extending sensory appendages to taste the water ahead and guide the colony toward the most promising areas. This division of labor created efficiencies that compounded over time. A feeding specialist could become extraordinarily good at extracting nutrients from the environment better than any generalist cell could ever be. A swimming specialist could develop propulsion systems that were faster and more energy efficient than anything a multitasking individual could maintain. The result was colonies that could find food faster, extract it more efficiently, and move to new locations with greater speed and precision. The benefits extended beyond just survival and feeding. Colonial life opened up entirely new ecological niches that had been inaccessible to individual cells. Large colonies could tackle food sources that were too big for single cells to handle. They could work together to break down complex organic molecules, or coordinate their efforts to overwhelm the defenses of other microorganisms. They could also survive in environments that would be fatal to isolated cells. Some locations in the ancient ocean had extreme conditions, high acidity, unusual mineral concentrations, or rapid temperature fluctuations that individual cells couldn't tolerate for long. But colonies could develop protective outer layers of specialized cells that shielded more sensitive interior members, creating their own microenvironments within the larger hostile world. Perhaps most intriguingly, colonial life began to make possible something that would eventually revolutionize life on Earth: the development of different cell types within the same organism. Early colonies probably started with all cells being essentially identical, but over time, the advantages of specialization became clear. Cells that were particularly good at one function could focus entirely on that role, while their partners handled other necessary tasks. This was the beginning of what scientists call differentiation, the process by which identical cells develop into different types with distinct functions. A feeding cell could gradually lose its swimming appendages and develop enhanced nutrient processing machinery. A protective cell could develop thicker membranes and defensive chemicals. A reproductive cell could focus entirely on the complex process of creating the next generation. The genetic instructions for this specialization were already present in every cell. After all, they were all descended from the same parent and carried identical DNA. But colonial life created the context where different genes could be activated in different cells, allowing the same genetic toolkit to produce radically different outcomes depending on each cell's role in the community. This innovation would eventually lead to the incredible diversity of cell types in complex organisms like yourself: muscle cells optimized for contraction, nerve cells specialized for rapid communication, skin cells designed for protection and barrier function. But it all began with those first simple colonies, where cells discovered that being different from their neighbors could benefit everyone. Let's rest here for a moment and marvel at this: the birth of us and we, in a universe that had only known I. Take a slow, deep breath and let yourself float back to that ancient ocean where we've been traveling together. Feel the gentle rhythm of primordial waves. Imagine the soft blue-green light filtering down through waters that had never known the shadow of a swimming fish or the song of a whale. In this peaceful setting, we've witnessed something that might seem small: just two cells choosing to remain connected after division instead of drifting apart into solitude. But oh, what a momentous choice it was. In that simple act of staying together, life discovered one of its most powerful secrets: that cooperation could be more successful than competition, that we could accomplish what I never could alone. Think about the profound shift this represented. For millions of years, every organism on Earth had been an island unto itself, complete, self-sufficient, but also completely alone. Success was measured purely in individual terms. How well can I survive? How effectively can I reproduce? How successfully can I pass on my genes to the next generation? But with the invention of colonial life, those questions suddenly became collective. How well can we survive together? How can we help each other reproduce more successfully? How can we ensure that our shared genetic legacy thrives? It was the universe's first experiment in community, the dawn of social organization, the beginning of every partnership and friendship and family that would ever exist. The molecular mechanisms we explored, those elegant cell adhesion proteins working like cosmic Velcro, the chemical communication systems that allowed cells to whisper secrets to each other, the mechanical forces that coordinated movement and growth, these were evolution's toolkit for building togetherness. They represent some of the most sophisticated engineering solutions ever developed. Systems so successful that you carry refined versions of them in every tissue of your body. We saw how these first partnerships were tentative and experimental, like shy dancers learning to move in harmony. Many failed, their molecular conversations breaking down under the stress of coordination. But the successful colonies discovered benefits that transformed life itself: better protection from predators, more efficient resource gathering, and the revolutionary possibility of cellular specialization. This last innovation, the idea that identical cells could take on different roles within the same organism, was perhaps the most important of all. It opened the door to complexity in ways that would have been impossible in a world of solitary cells. It meant that life could begin experimenting with division of labor, with efficiency through specialization, with the kind of coordinated teamwork that would eventually build hearts and brains and eyes. As you lie there now, your own body is a testament to the success of that ancient experiment in cooperation. Your heart cells are beating in perfect synchrony, not because they have to, but because they've chosen to work together for the benefit of the whole. Your brain cells are firing in coordinated patterns that create thoughts and memories and dreams. Your immune cells are standing guard, protecting the community of your body from threats, just as those first colonial cells protected each other in the primordial seas. The thread that connects you to those ancient pioneers is unbroken, a chain of cooperation stretching back nearly 1 billion years, each generation building on the innovations of the last, each organism a community of cells that have chosen togetherness over isolation. In our next chapter, we'll explore how this simple partnership between cells evolved into something even more remarkable: the development of specialized roles and the birth of the first true animal bodies. We'll see how division of labor led to the invention of tissues and organs, and how cooperation became so sophisticated that individual cells began to sacrifice their own independence for the good of the whole. But for now, rest in the wonder of this moment we've shared, the moment when life discovered that it was better together than apart, when the universe learned to say we instead of just I. Feel the ancient wisdom of cooperation flowing through your own cells, the same molecular wisdom that first stirred in those primordial waters so long ago. The journey from one to many, from individual to community, from I to we, this is not just the story of evolution. It's the story of every friendship you've ever formed, every family that's ever gathered, every community that's ever come together to accomplish something greater than any individual could achieve alone. And it all began with two tiny cells in an ancient ocean, making the simple but revolutionary choice to stay together. As these cellular communities grew more comfortable with togetherness and comfort, in the context of evolution, is measured not in days or months but in millions of years of gradual refinement. Something even more remarkable began to happen in the ancient seas. The early colonies we met in our last chapter, those tentative partnerships of 2 or 4 or 8 identical cells, were about to discover one of life's most powerful innovations, the art of being different on purpose. Picture, if you will, a small colony drifting through those primordial waters, its member cells still fundamentally identical but beginning to sense new possibilities stirring in their molecular conversations. They had mastered the basic skills of staying together, of coordinating their movements, of sharing resources and information. But now, after countless generations of successful cooperation, a new kind of whisper was beginning to pass between them. Not just the simple chemical signals that said, "I'm here," or "food this way," but something more sophisticated, more purposeful. Some cells began to whisper to others, "What if you handled the food, and I'll watch for danger?" It was a revolutionary idea, though of course no conscious thought was involved. Instead, it emerged from the patient logic of natural selection, as colonies that experimented with task division found themselves more successful than those where every cell tried to do everything. The transition was gradual, almost imperceptible at first. In one colony, a cell that happened to be particularly good at detecting nutrients might find itself spending more and more of its energy on that activity. Its molecular machinery, through countless tiny adjustments, would begin to optimize for feeding, developing more sensitive chemical receptors, more efficient nutrient processing systems, more specialized transport mechanisms. Meanwhile, its neighbor, perhaps positioned at the edge of the colony where it was more likely to encounter threats, might find its cellular resources gradually shifting toward vigilance and protection. Its membrane sensors would become more attuned to the chemical signatures of danger, its internal chemistry more prepared for rapid defensive responses. This process was like watching a jazz ensemble find its groove, with each musician gradually settling into their unique role, not through explicit planning, but through the natural dynamics of creative collaboration. Except this musical improvisation was happening at the molecular level, with genes switching on and off, proteins changing their expression patterns, and cellular architectures slowly reshaping themselves in response to their new specialized functions. The beauty of this process lies in its elegant simplicity. All the cells in a colony still carried identical DNA, the same genetic instruction manual that had been copied faithfully from their common ancestor. But genes, like the pages in a vast library, don't all need to be read at once. Different cells could focus on different chapters of their genetic book, expressing some genes strongly while leaving others silent, creating remarkable diversity from identical starting points. It was as if each cell had access to the same comprehensive toolkit, but chose to become expert with different tools. One cell might specialize in the molecular equivalents of hammers and nails, becoming a master builder of structural proteins. Another might focus on the chemical equivalents of sensors and alarms, developing exquisite sensitivity to environmental changes. A third might become like a molecular chef, specializing in the complex recipes needed to break down and process different types of food. The environmental pressures that encouraged this specialization were becoming more intense with each passing geological epoch. The ancient ocean was growing more crowded as life diversified and multiplied. Competition for resources was intensifying, predation was becoming more sophisticated, and the simple strategies that had worked for solitary cells were no longer sufficient in this more complex world. Colonies that could divide labor had decisive advantages. They could respond to multiple challenges, simultaneously feeding while watching for danger, reproducing while maintaining their structure, exploring new territories while defending their current position. They became more efficient, more resilient, more adaptable to changing conditions. But perhaps most importantly, specialization opened the door to capabilities that would have been impossible for generalist cells. A cell that devoted all its resources to swimming could develop propulsion systems far more powerful than anything a multitasking individual could maintain. A cell that focused entirely on defense could produce protective compounds at concentrations that would be toxic to a cell trying to perform other functions. A cell specialized for reproduction could develop the complex machinery needed for sophisticated genetic recombination and developmental control. Like members of a tiny orchestra, each cell began to find its unique voice in the growing symphony of multicellular life. And what a miraculous orchestra it was. No conductor waving a baton, no sheet music to follow, just the natural harmony that emerged when individual excellence served collective success. The process of cellular specialization, which scientists would eventually call differentiation, was one of evolution's most elegant solutions to the challenge of complexity. Imagine having access to an enormous library containing all the knowledge needed to build and maintain life, but instead of everyone trying to read every book at once, which would be overwhelming and inefficient, different individuals focus on becoming experts in different subjects. This is essentially what happened as cells learned to specialize. Each cell in a colony carried the complete genetic library, containing instructions for every possible cellular function. But through intricate molecular switches and regulatory systems, different cells could choose which parts of this library to read and which to keep closed. The mechanisms behind this selection process were works of art in themselves. Special regulatory proteins, like molecular librarians, would patrol the DNA and decide which genes should be actively transcribed at any given time. Some of these proteins responded to chemical signals from neighboring cells, essentially allowing the community to vote on what roles each member should play. Others responded to the cell's position in the colony, creating different types of specialists based on whether a cell was on the inside or outside, at the front or back, at the top or bottom of the group. Environmental cues also played crucial roles in determining cellular fate. A cell exposed to higher concentrations of nutrients might be guided toward a feeding specialization, while one experiencing more mechanical stress might develop into a structural support cell. The system was beautifully responsive, allowing colonies to adapt their internal organization to match external conditions. The first feeding specialists developed molecular machinery that would make a modern chemist envious. They created arrays of receptor proteins that could detect and bind specific nutrients from the surrounding water with extraordinary sensitivity and selectivity. Their internal chemistry became optimized for rapid processing of these captured molecules, breaking them down into simpler components that could be shared with other cells in the colony through increasingly sophisticated distribution networks. These feeding cells also developed storage systems, becoming like the pantries of their colonies. They could stockpile nutrients during times of abundance and release them gradually during lean periods, providing stability and security for the entire community. Some even began to specialize in processing specific types of food. One cell type might focus on simple sugars, another on proteins, still another on lipids, creating a diverse metabolic ecosystem within each colony. The first structural specialists took on the role of architects and engineers, developing enhanced adhesion systems and beginning to produce extracellular materials that could provide additional support and organization to the colony. They created molecular scaffolding that helped maintain the colony's shape during movement and growth, and they developed the ability to repair damage when parts of the structure were injured or lost. Perhaps most remarkably, some structural cells began to produce materials that would become the foundation for more complex body organization. They secreted proteins that could form organized matrices, creating the first primitive versions of what would eventually become connective tissues, cartilage, and bone. These materials provided not just support but also created new microenvironments within the colony, specialized niches where other types of cells could develop and thrive. The first motility specialists transformed themselves into living engines, developing enhanced flagella, cilia, and other propulsion systems that could move entire colonies through the water with unprecedented speed and precision. These cells often clustered together in specific regions of the colony, creating the first primitive organs dedicated to locomotion. Some motility cells became incredibly sophisticated, developing the ability to change their beating patterns in response to chemical gradients or mechanical forces. They could steer the colony toward favorable conditions and away from threats, essentially becoming the navigation systems for these microscopic voyagers. The first sensory specialists developed into the eyes and ears of their colonies. though of course these early sensors were far simpler than the organs we associate with those terms. They created specialized receptor systems that could detect changes in light, chemical composition, temperature, or mechanical forces with remarkable sensitivity. These sensory cells often developed internal signal amplification systems, allowing them to detect incredibly faint stimuli and transmit information about environmental conditions to other members of the colony. They became the early warning systems that could alert their communities to approaching dangers or opportunities. Perhaps most intriguingly, some sensory specialists began to develop primitive information processing capabilities. Instead of simply detecting stimuli and passing along raw data, they started to interpret and integrate information from multiple sources, making the first tentative steps toward what would eventually become nervous systems. The coordination of all these specialists required increasingly sophisticated communication systems. Chemical signaling networks became more complex, with different molecular messengers carrying different types of information. Some signals were local, affecting only immediate neighbors, while others could propagate throughout the entire colony, coordinating colony-wide responses to changing conditions. Physical connections between cells also became more specialized. Simple adhesion proteins evolved into more complex junction systems that could control the flow of materials and information between cells. Some junctions allowed free exchange of small molecules, maintaining biochemical unity across the colony. Others were more selective, creating barriers that allowed different regions to maintain distinct internal environments. And slowly, almost imperceptibly, something new was taking shape in the ancient waters, the very first animal bodies. What had begun as simple clusters of cooperating cells was evolving into something far more sophisticated and organized structures that we would recognize as the earliest ancestors of every animal that has ever lived, including you. The transition from colonial clusters to true multicellular bodies was gradual and beautiful, like watching a flower bloom in extreme slow motion across geological time. The key innovation was the development of spatial organization, the ability to create consistent, reproducible arrangements of different cell types that could work together as integrated systems. Early colonies had been somewhat haphazard in their organization. Specialized cells might cluster together in loose groups, but there was no consistent pattern, no reliable architecture that could be passed on from one generation to the next. The breakthrough came when genetic control systems evolved that could specify not just what type of cell to become, but where in the developing organism that cell type should be located. This spatial control system was like having a molecular blueprint that could guide the construction of a living building. It specified which types of cells should form the foundation, which should create the walls, which should become the roof. It ensured that feeding cells would be positioned where they could access nutrients most effectively, that structural cells would be arranged to provide optimal support, that sensory cells would be placed where they could best monitor the environment. The first organized body plans were elegantly simple. Picture a hollow sphere, its wall composed of a single layer of cells, drifting through the primordial seas like a microscopic balloon. This basic architecture, which scientists would eventually call a blastula, solved several important problems simultaneously. It created a clear distinction between inside and outside, allowing the organism to control its internal environment while maintaining contact with the external world. It maximized the surface area available for nutrient absorption and waste disposal, and it provided a stable, symmetric structure that could move efficiently through the water. But even this simple organization opened up new evolutionary possibilities. Different regions of the sphere could begin to specialize in different functions. One hemisphere might become particularly good at swimming, while the opposite pole might focus on feeding. The equatorial region might develop enhanced sensory capabilities, creating a band of specialized cells that could monitor the environment in all directions. The next great innovation was even more profound: the development of two distinct cell layers, creating organisms with both an outer surface and an inner cavity. This advance, which biologists call gastrulation, was revolutionary because it established the fundamental architectural principle that would guide animal body plan development for the next 600 million years. Imagine our hollow sphere beginning to fold in on itself, like a balloon being pushed inward from one side. The result would be a double-walled structure with an outer layer in contact with the environment and an inner layer lining a central cavity. This inner cavity could become specialized for digestion, creating the first primitive gut. while the outer layer could focus on protection, movement, and environmental sensing. This two-layered organization, with its distinction between outer ectoderm and inner endoderm, provided a whole new level of functional specialization. The outer layer could develop into skin, nerve tissue, and sensory organs, all the systems needed to interact with the external world. The inner layer could specialize in digestion, nutrient processing, and waste management, all the internal housekeeping functions that keep a complex organism running. The cavity between these layers, which would eventually become the body cavity of more complex animals, created space for the development of internal organs and systems. It provided room for specialized structures like primitive hearts, simple filtering systems, and storage organs. It also created new opportunities for internal transport, as fluid in the cavity could carry nutrients, waste products, and signaling molecules throughout the organism. The genetic control systems that guided this organized development were marvels of molecular engineering. They involved networks of regulatory genes that could be switched on and off in precise sequences, creating waves of cellular differentiation that swept through the developing organism in carefully orchestrated patterns. Some of these control genes, remarkably, are still active in your own body today. The molecular switches that determined where head and tail, front and back, left and right would form in those first organized bodies have been conserved and elaborated over hundreds of millions of years of evolution. The same basic genetic toolkit that built the first simple body plans is still building bodies today, though it has been refined and expanded to create the incredible diversity of animal forms we see in the modern world. The development of organized body plans also required sophisticated cell-to-cell communication systems. Cells needed to know not just what to become, but when to change and how to coordinate their transformations with their neighbors. This led to the evolution of complex signaling pathways that could carry positional information throughout the developing organism. Some signals were like molecular postal codes, telling cells where they were located in the body plan. Others were like timing signals, coordinating the sequence of developmental events. Still others carried information about environmental conditions, allowing the developing organism to adjust its body plan in response to external circumstances. It was in this context of increasing organization and sophistication that one of evolution's most successful early experiments emerged, the cnidarians, that remarkable group of animals that includes modern jellyfish, sea anemones, corals, and hydra. These creatures represent a pivotal moment in animal evolution, and their story illuminates one of the most profound transitions in the history of life on Earth. Imagine the first creatures to have both a mouth and a way of moving through water with purpose. This might seem like a simple combination to us, accustomed as we are to animals that can chase their food and eat it efficiently. But in the ancient seas, this was a revolutionary innovation that opened up entirely new ways of living. The cnidarians were among the first animals to develop true tissues, organized groups of specialized cells that could work together to perform specific functions. Unlike the loose colonies we've been discussing, cnidarian bodies were integrated systems where different tissue types were arranged in precise, reproducible patterns that could be reliably transmitted from one generation to the next. Their body plan was elegantly simple yet remarkably effective. Picture a living cup or bell, its walls made of two distinct tissue layers. The outer layer, called the epidermis, was specialized for protection, movement, and environmental sensing. The inner layer, the gastrodermis, was optimized for digestion and nutrient processing. Between these layers was a simple but effective support structure called the mesoglia, a jelly-like material that provided both structural integrity and buoyancy. This basic architecture could be modified in endless ways to suit different lifestyles. In jellyfish, the bell became a sophisticated propulsion system, contracting rhythmically to push water and propel the animal through the sea with graceful, pulsing movements. In sea anemones, the body stretched into a column, topped with a crown of tentacles, creating a sessile predator that could capture prey while remaining anchored to the seafloor. In corals, individual bodies could join together in colonies, creating reef systems that would become some of the most diverse ecosystems on Earth. But perhaps the most remarkable innovation of the cnidarians was their development of the first true nervous system. This was a quantum leap in biological organization, the evolution of specialized cells that could rapidly transmit information throughout the body, coordinating responses to environmental stimuli with unprecedented speed and precision. The cnidarian nervous system was elegantly simple, organized as a nerve net rather than the centralized nervous systems we see in more complex animals. Imagine a delicate web of interconnected nerve cells spread throughout the body, like a living internet that could carry electrical signals from one region to another. This network allowed different parts of the animal to communicate and coordinate their activities in real time. When a tentacle detected prey, for example, nerve signals could rapidly spread throughout the body, triggering coordinated responses. Other tentacles would help capture the prey, the mouth would prepare for feeding, and the entire body might adjust its position to optimize the feeding process. This level of coordination required split-second timing and system-wide communication that would have been impossible with purely chemical signaling. The nerve cells themselves were marvels of bioengineering. They developed the ability to generate and propagate electrical signals using carefully controlled movements of ions across their membranes to create waves of electrical activity that could travel at remarkable speeds. They also developed specialized connections called synapses, where electrical signals could be converted to chemical signals and back again, allowing for complex processing and integration of information. Perhaps most remarkably, cnidarians also developed the first muscle cells, specialized tissues that could contract forcefully and rapidly in response to nerve signals. These muscle cells made possible the coordinated movements that we associate with animal behavior. The pulsing of a jellyfish bell, the closing of a sea anemone when threatened, the coordinated beating of cilia that could create water currents for feeding. The muscle cells were integrated with the nervous system in sophisticated ways. Different types of muscle could be controlled independently, allowing for complex patterns of movement. Some muscles were designed for rapid, powerful contractions that could propel the animal through water or capture prey. Others were optimized for sustained contractions that could maintain body posture or create steady water currents. The cnidarians also developed another innovation that would prove crucial for animal evolution: specialized stinging cells called cnidocytes. These remarkable structures were like microscopic harpoons, capable of explosively firing toxic barbs into prey or predators. They represented one of the first examples of sophisticated cellular weaponry, and they made cnidarians formidable predators despite their relatively simple body organization. The stinging cells were integrated into the overall body plan in elegant ways. They were concentrated in the tentacles and around the mouth, where they would be most effective for prey capture. They could be triggered by both chemical and mechanical stimuli, allowing the animal to distinguish between food and non-food objects. And they could be controlled by the nervous system, allowing for coordinated attacks when large prey was detected. The success of the cnidarian body plan is evidenced by its persistence over hundreds of millions of years. Modern jellyfish are remarkably similar to their ancient ancestors, testimony to the effectiveness of this early solution to the challenges of multicellular life. They have survived mass extinctions, dramatic climate changes, and the evolution of countless more complex predators and competitors. Their influence on subsequent animal evolution was profound. The tissue-level organization pioneered by cnidarians became the foundation for all later animal development. The nerve muscle systems they developed were elaborated and refined in countless ways, eventually giving rise to the sophisticated nervous systems and muscular systems of modern animals. Even their simple gut cavity provided the architectural foundation for the complex digestive systems of more advanced creatures. In your own body right now, billions of cells are cooperating in ways that would make those ancient pioneers proud. As you lie there, breathing softly, perhaps feeling the gentle rhythm of your heartbeat, you are experiencing the culmination of nearly 1 billion years of evolutionary refinement in cellular cooperation and specialization. Each cell in your heart has chosen to beat in rhythm with its neighbors, a dance that began nearly 1 billion years ago in those first tentative partnerships we've been exploring together. The cardiac muscle cells that make up your heart wall are descendants of those early muscle specialists, but they have been refined over countless generations into precision instruments of synchronized contraction. Your heart cells don't just beat together, they communicate constantly through specialized connections called gap junctions, sharing electrical signals that ensure perfect timing across the entire organ. When one cell begins to contract, it sends a wave of electrical activity to its neighbors, creating the coordinated rhythm that has been pumping steadily in your chest since before you were born. But the cooperation goes far deeper than just synchronized beating. Your heart muscle cells have developed the most sophisticated energy production systems of any cells in your body, packed with mitochondria that work around the clock to generate the ATP needed for continuous contraction. They have specialized protein structures that can convert electrical signals into mechanical force with extraordinary efficiency. They even have their own repair systems that can maintain and rebuild the cellular machinery throughout your lifetime. The nervous system that controls your heart represents another stunning example of cellular cooperation and specialization. The nerve cells that regulate your heartbeat are themselves the products of millions of years of refinement of those first simple nerve nets we encountered in the cyanidarians. They can integrate information from throughout your body, your activity level, your emotional state, your need for oxygen, and adjust your heart rate accordingly, all without any conscious effort on your part. Your brain, as you listen to these words and form thoughts about ancient cellular partnerships, is perhaps the most remarkable example of specialized cooperation in the known universe. The 100 billion neurons in your brain are each in communication with thousands of others, creating a network of incredible complexity and sophistication that emerges from the same basic principles of cellular cooperation that first appeared in those primordial seas. Each neuron in your brain is a specialist, but unlike the early cellular specialists we've been discussing, these cells have developed the ability to process information, to receive signals, integrate them, and produce responses that can influence the behavior of other neurons. They have created patterns of connectivity that can store memories, generate emotions, solve problems, and even contemplate their own existence. The cooperation between your neurons is so seamless that you experience it as unified consciousness, as a single stream of awareness flowing through time. But in reality, every thought you think, every memory you recall, every decision you make emerges from the coordinated activity of millions of specialized cells, each contributing its small part to the larger symphony of your mental life. Your digestive system represents yet another triumph of cellular specialization and cooperation. The cells lining your stomach have become expert chemists, producing powerful acids and enzymes that can break down the most complex foods. The cells in your small intestine have developed intricate absorption systems that can extract nutrients with remarkable efficiency and selectivity. The cells in your liver perform hundreds of different chemical transformations, detoxifying harmful substances while producing essential proteins and other molecules. All of these systems work together with a level of coordination that would be impossible without the sophisticated communication networks that connect every part of your body. Chemical messengers called hormones carry information through your bloodstream, allowing distant organs to coordinate their activities. Your nervous system provides rapid communication pathways that can integrate responses across your entire body in milliseconds. Even your immune system represents a remarkable example of cellular cooperation and specialization. Different types of immune cells patrol your body, each with specialized functions. Some are scouts that detect invading pathogens, others are assassins that destroy infected cells. Still others are memory specialists that remember past encounters and prepare for future threats. They communicate through complex networks of chemical signals, creating coordinated responses that can protect you from countless different types of danger. The skin cells that protect your body's outer surface represent perhaps the ultimate expression of cellular self-sacrifice for the greater good. These cells are programmed to die, creating layers of protective material that shield the living cells beneath from environmental damage. They replace themselves constantly, maintaining the integrity of your body's boundary with the outside world through the ultimate act of cellular cooperation, giving up their own lives for the benefit of the community. As you breathe in and out, your lung cells are performing another ancient dance of cooperation. The cells lining your airways work together to keep the passages clean and moist. The cells in your lungs' tiny air sacs have become incredibly thin and permeable, optimized for the efficient exchange of oxygen and carbon dioxide. They coordinate with your red blood cells themselves, highly specialized cells that have given up their nuclei to become more efficient oxygen carriers, to maintain the delicate balance of gases that keeps every other cell in your body alive. This is the legacy of that first moment when two cells chose to stay together rather than drift apart in the ancient seas. Every beat of your heart, every breath you take, every thought that crosses your mind is a testament to the power of cooperation, the beauty of specialization, and the profound success of the multicellular experiment that began so long ago. The next time you feel your pulse, remember that you are experiencing an unbroken chain of cellular cooperation stretching back nearly 1 billion years. The same molecular mechanisms that allowed those first simple colonies to coordinate their activities are still coordinating the activities of your trillions of cells, refined and elaborated, but fundamentally unchanged in their commitment to working together for the benefit of the whole. You are not just made of cooperating cells, you are cooperation itself, given form and consciousness. You are the universe's way of experiencing its own creativity, its own capacity for organization and complexity and beauty. You are living proof that togetherness is more powerful than separation, that specialization serves cooperation, and that the whole can indeed be greater than the sum of its parts. In our next chapter, we'll explore how these principles of cooperation and specialization were taken to even greater heights, as life began to elaborate and diversify into the incredible variety of animal forms that would populate the Earth. We'll witness the great explosion of creativity that would fill the oceans with creatures of unprecedented complexity and beauty, each one a unique solution to the challenges of multicellular life. But for now, rest in the awareness of your own cellular community, marveling at the ancient wisdom that flows through every part of your being, the billion-year-old conversation between cooperation and specialization that has created the miracle of you. Around 540 million years ago, something extraordinary happened in the story of life on Earth. Life seemed to wake up and remember it had been dreaming. After nearly 4 billion years of mostly microscopic existence, after hundreds of millions of years of patient experimentation with cellular cooperation and simple body plans, the stage was set for one of the most remarkable episodes in the history of our planet. Picture, if you will, the ancient seas at the dawn of this new era. The water was clearer than it had been in eons, as complex ecosystems had evolved to filter and process nutrients with unprecedented efficiency. The chemistry of the ocean itself had changed, with oxygen levels rising to support more energetic forms of life, while mineral compositions had stabilized in ways that made building shells and hard parts not just possible, but advantageous. The temperature was moderate and stable, creating vast, shallow seas that served as nurseries for evolutionary innovation. Ocean currents carried nutrients and genetic material across great distances, allowing successful innovations to spread and intermingle. And perhaps most importantly, the basic toolkit for building complex bodies, the genetic machinery, the cellular communication systems, the fundamental architectural principles we explored in our previous chapters, had been tested and refined over countless generations. It was like a master chef finally having access to the finest ingredients, the perfect kitchen, and centuries of accumulated culinary wisdom all at the same moment. The results were spectacular. This period, which scientists would eventually call the Cambrian explosion, was not actually an explosion in the sense of a sudden, violent event. Rather, it was more like a grand flowering, a gentle but persistent blooming of biological creativity that unfolded over millions of years, rapid by geological standards, but still gradual enough that each innovation could be thoroughly tested and refined before the next breakthrough emerged. During this remarkable time, virtually every major body plan that exists in the animal kingdom today made its first appearance in the fossil record. It was as if life was conducting a vast experiment in form and function, trying out every possible way that cells could organize themselves into complex, multicellular bodies. Some of these experiments were so successful that they have persisted virtually unchanged for 500 million years. Others were brief but brilliant innovations that pushed the boundaries of what was possible before giving way to even more sophisticated designs. The whisper that announced this great elaboration was subtle. At first, slightly larger creatures appearing in ancient sediments, new types of burrows showing up in seafloor deposits, the first hints of hard shells and complex appendages preserved in stone. But as millions of years passed, the whisper became a chorus, then a symphony, as the seas filled with an unprecedented diversity of forms. What made this explosion of creativity possible was the convergence of several crucial factors. The genetic toolkit for building complex bodies had reached a level of sophistication that could support elaborate developmental programs. Environmental conditions had stabilized in ways that rewarded size and complexity. And perhaps most importantly, the ecosystem itself had reached a level of maturity where there were numerous ecological niches waiting to be filled by creatures with the right combination of adaptations. It was during this time that predation became a serious evolutionary force. The appearance of animals with sophisticated sensory systems, rapid locomotion, and effective weapons created intense pressure for defensive innovations. The result was an evolutionary arms race that drove increasing complexity and sophistication in both predators and prey. The Cambrian seas were unlike anything that had existed before or has existed since. They were filled with creatures that seem almost fantastical to our modern eyes, arthropods with dozens of legs and elaborate compound eyes, strange worm-like animals with spines and armor plating, early mollusks experimenting with the first spiral shells, primitive chordates developing the earliest versions of what would eventually become backbones. Many of these early experiments in complex life have no modern equivalents. They represent evolutionary paths that were explored briefly before being abandoned in favor of more successful designs. But studying their fossilized remains gives us glimpses into the incredible creativity and flexibility of the evolutionary process during this remarkable period. The pace of innovation during the Cambrian was breathtaking. New body plans appeared, were tested, refined, and sometimes discarded in what amounts to the blink of an eye in evolutionary terms. It was as if life was in a hurry to explore every possible variation on the theme of multicellular organization, driven by both opportunity and necessity to find the most effective solutions to the challenges of complex existence. Our single-celled ancestor had passed down something precious through all those eons of evolution. A toolkit for building bodies that was far more sophisticated and versatile than anyone could have imagined. This toolkit, refined and expanded over hundreds of millions of years of cellular cooperation and specialization, was about to prove capable of building forms of almost unimaginable complexity and beauty. Like a master craftsperson's workshop, passed down through generations and continually refined with new tools and techniques, the genetic machinery of life had become extraordinarily sophisticated by the time of the Cambrian explosion. At the heart of this toolkit were special genes that scientists would eventually call Hox genes, molecular architects, that could specify not just what types of cells to build, but where in the developing body they should be located. Imagine having a set of molecular blueprints so elegant and flexible that they could be used to build everything from a simple worm to a complex arthropod to the basic body plan that would eventually give rise to vertebrates like ourselves. These Hox genes were like master switches that could activate whole cascades of developmental processes, turning on hundreds of other genes in precisely coordinated sequences. The beauty of the Hox gene system lies in its modular organization. Different Hox genes control the development of different body segments, like chapters in a book that could be read independently or combined in various ways to create different stories. A creature could have a simple body plan with just a few repeated segments, or a complex one with many different specialized regions, all controlled by variations in how these master control genes were expressed. What's truly remarkable is that the same basic Hox genes that were directing body plan development during the Cambrian explosion are still active in developing animals today. The molecular switches that determined head from tail, front from back, and the overall organization of body segments in those ancient seas are the same ones that helped shape your own body during your development in your mother's womb. But the toolkit extended far beyond just the Hox genes. There were genes for building eyes, genes for constructing shells and armor, genes for developing complex appendages, genes for creating sophisticated nervous systems. Each of these genetic programs could be mixed and matched, combined in different ways, and activated at different times during development to create an enormous variety of body forms. The signaling systems that allowed cells to communicate during development had also become incredibly sophisticated. Chemical messengers could carry positional information throughout the developing embryo, telling cells not just what to become, but when to change and how to coordinate their transformations with their neighbors. Some signals worked over long distances, creating gradients that could specify body axes and overall organization. Others worked locally, fine-tuning the development of specific tissues and organs. The cellular machinery for interpreting these signals had evolved to remarkable levels of precision and reliability. Cells could integrate information from multiple sources, making complex decisions about their fate based on their position, their neighbors, their internal state, and environmental conditions. They could switch between different developmental programs, create backup systems in case of damage, and even adjust their development in response to changing circumstances. Perhaps most impressively, the toolkit had developed sophisticated quality control systems. Developing organisms could detect errors in their body, plan construction, and either correct them or eliminate defective cells before they could cause problems. This reliability was crucial for building the larger, more complex bodies that were becoming possible during the Cambrian. The toolkit also included increasingly sophisticated systems for building specialized structures. The genetic programs for constructing eyes, for example, had become so refined that they could create complex optical systems with lenses, focusing mechanisms, and sophisticated neural processing networks. The programs for building shells could produce structures of incredible strength and beauty, with complex spiral geometries and layered construction that optimized both protection and efficiency. Muscle-building programs had evolved to create movement systems of unprecedented power and precision. Some creatures developed rapid escape responses that could propel them away from danger in milliseconds. Others developed precise manipulation abilities that allowed them to handle food and construction materials with remarkable dexterity. The nervous system construction programs had become capable of building information processing networks that could support complex behaviors. Simple reflex circuits had evolved into sophisticated neural networks that could learn, remember, and make decisions based on past experience and current conditions. All of these genetic programs were modular and combinatorial, meaning they could be mixed and matched in countless different ways to create new body plans and new capabilities. It was like having a vast library of biological subroutines that could be called upon and combined to solve whatever challenges evolution presented. The flexibility of this system meant that evolutionary innovation could happen remarkably quickly. A successful predator adaptation in one lineage could drive defensive innovations in prey species, which could in turn drive new predator adaptations, creating rapid evolutionary change that filled the seas with increasingly sophisticated forms. During this remarkable period of biological creativity, life experimented with innovations that would seem like science fiction if we didn't have the fossil evidence to prove they actually existed. Each new innovation opened up new possibilities, new ecological niches, new ways of interacting with the environment and with other creatures. Perhaps no innovation during the Cambrian explosion was more revolutionary than the development of sophisticated vision systems. Before this time, light had been nothing more than a source of energy or a simple environmental cue. But suddenly, creatures began to develop the ability to form images, to see their world in detail, to track prey and avoid predators with unprecedented precision. The first eyes were probably simple, light-detecting patches, no more complex than the eye spots we still see in some single-celled organisms today. But the advantages of being able to detect the direction and intensity of light were so great that natural selection rapidly refined these simple structures into increasingly sophisticated optical systems. Some creatures developed compound eyes, with hundreds or even thousands of individual optical units called ommatidia, each one a complete light-detecting system with its own lens and photoreceptor cells. These compound eyes could detect the slightest movements across a wide field of view, making them perfect for spotting predators or prey. The mathematical precision of their construction rivals anything we can build with modern engineering hexagonal arrays of lenses that maximize coverage while minimizing optical distortion. Other creatures experimented with simple eyes, developing single large lenses that could focus light onto sensitive retinas. These eyes could form sharp images and detect fine details, perfect for examining food items or navigating complex environments. The optical principles they employed, the physics of refraction and focus, are the same ones we use in our cameras and telescopes today. But perhaps most remarkably, many different lineages of animals independently invented eyes during this period. Arthropods developed their compound eyes, mollusks created their camera-like eyes, and early chordates began experimenting with the light-sensitive patches that would eventually become vertebrate eyes. It was as if the universe itself was eager to see, driving evolution to discover vision again and again through different pathways. The development of sophisticated vision systems had profound effects that rippled throughout the entire ecosystem. Predators could hunt more effectively, but prey could also detect threats from greater distances. The result was an escalating evolutionary arms race that drove improvements in speed, agility, camouflage, and defensive structures. Vision also enabled entirely new types of behavior. Creatures could begin to recognize each other as individuals, laying the groundwork for more complex social interactions. They could navigate using visual landmarks, explore their environments more effectively, and engage in sophisticated foraging behaviors that required detailed visual information about their surroundings. The neural systems required to process visual information also drove advances in brain development. Creating detailed images from the raw data provided by photoreceptor cells required sophisticated neural networks that could integrate information, detect patterns, and extract meaningful features from complex visual scenes. These advances in neural processing would prove crucial for many other aspects of animal behavior and cognition. Another revolutionary innovation of the Cambrian explosion was the development of hard external structures, shells, carapaces, spines, and armor plating that could provide protection while still allowing for growth, movement, and feeding. This was essentially the invention of architecture in the biological world, the first time creatures learned to build and carry their own houses. The chemistry of shell construction was remarkably sophisticated. Creatures learned to extract calcium carbonate, silica, and other minerals from seawater and deposit them in precisely controlled patterns to create structures that were both strong and lightweight. The resulting shells often had complex layered architectures that distributed stress efficiently and could resist both crushing forces and penetrating attacks. Some creatures developed spiral shells that grew continuously throughout their lives, maintaining perfect mathematical proportions based on logarithmic spirals that optimized both strength and internal space. Others created segmented armor that could flex and move while still providing protection, like biological chainmail that covered their entire bodies. The diversity of defensive strategies was extraordinary. Some creatures developed spines and projections that made them difficult to swallow. Others created thick, heavy armor that could withstand tremendous crushing forces. Still, others developed shells that could be quickly sealed shut, creating impregnable fortresses that could wait out attacks. But shells and armor weren't just passive defenses. Many creatures learned to use their hard parts as tools and weapons. Appendages could be reinforced with hard materials to create powerful claws and pincers. Body segments could be modified into ramming devices or cutting tools. Even shells could be weaponized, with some creatures developing the ability to rapidly seal their shells with explosive force, creating biological pile drivers. The ability to build hard structures also opened up new ecological niches. Creatures could now live in high-energy environments where wave action would have destroyed soft-bodied forms. They could exploit food sources that required powerful crushing or cutting tools to access. They could even modify their environments, with some early creatures beginning to build reefs and other structures that provided habitat for entire communities of organisms. Perhaps the most versatile innovation of the Cambrian explosion was the development of jointed appendages, arms, legs, antennae, and other projections that could extend the reach and capabilities of the basic body plan in countless different ways. This was the moment when bodies learned to reach out and actively manipulate their environment rather than simply responding to whatever came their way. The basic principle behind jointed appendages is elegantly simple. By connecting rigid segments with flexible joints, creatures could create structures that were both strong and highly maneuverable. Each joint could move in specific directions, and by coordinating the movements of multiple joints, complex three-dimensional motions became possible. Early arthropods became masters of appendage design, developing legs for walking, swimming paddles for locomotion through water, feeding appendages for capturing and processing food, sensory antennae for detecting chemical and mechanical stimuli, and gills for extracting oxygen from water. Many of these functions were combined in single appendages that could serve multiple purposes. A leg might also function as a gill and a feeding tool, maximizing efficiency while minimizing the energy cost of construction and maintenance. The diversity of appendage modifications was breathtaking. Some creatures developed dozens of nearly identical legs that could create powerful swimming motions or allow for rapid locomotion across the seafloor. Others specialized their appendages for specific functions, with some legs modified for digging, others for grasping, and still others for delicate manipulation of food items. Feeding appendages became particularly sophisticated, with some creatures developing complex arrays of specialized mouthparts that could handle different types of food. Filter-feeding appendages could strain tiny particles from the water with extraordinary efficiency. Predatory appendages could capture and dismember prey with surgical precision. Even scavenging appendages could sort through sediments to find buried food items. The sensory capabilities of appendages also advanced rapidly. Antennae could detect incredibly faint chemical signals, allowing creatures to track prey or find mates across great distances. Specialized appendages could detect water movements, pressure changes, and even electrical fields generated by other organisms. Perhaps most importantly, appendages enabled creatures to begin actively shaping their environment rather than simply adapting to it. They could dig burrows, build shelters, move obstacles, and even engage in primitive tool use. This was the beginning of what we might call technology in the biological world, the use of external objects and structures to extend natural capabilities. From that single cellular decision to stay together nearly 1 billion years earlier, an explosion of creativity was now unfolding that would establish the basic patterns of animal life for the next 500 million years. The ancient seas became filled with creatures that would seem fantastical to modern eyes, each one representing a unique solution to the challenges and opportunities of complex multicellular existence. The arthropods emerged as one of the most successful groups, their jointed bodies and specialized appendages proving incredibly versatile. Trilobites scuttled across the seafloor on dozens of legs, their compound eyes scanning for food and threats. Some grew to enormous sizes, becoming the first large predators in Earth's oceans. Others remained small and delicate, specializing in filtering tiny particles from the water or burrowing through sediments. The early arthropods experimented with body plans that have no modern equivalents. Some had bodies with dozens of segments, each bearing a different type of appendage specialized for a particular function. Others had bizarre head regions with multiple pairs of grasping arms and complex feeding apparatus that could process a wide variety of food types. Opabinia, one of the strangest creatures from this period, had 5 mushroom-shaped eyes on its head and a long, flexible proboscis ending in a claw-like grasping organ. It moved through the water with undulating fins along its sides, using its bizarre feeding appendage to pluck prey from the sea floor. No creature like it exists today, but it represents one of evolution's creative experiments in combining mobility, vision, and predation in a single body plan. The mollusks were also innovating rapidly during this period, developing the first sophisticated shells and experimenting with different body organizations. Some early mollusks were flat and crawled along the seafloor like living pancakes. Others developed tall, cone-shaped shells that they could retreat into when threatened. Still, others began experimenting with coiled shells that would eventually give rise to the spectacular diversity of snails and nautiloids. Early cephalopods appeared during this time, developing the ability to move by jet propulsion and beginning to experiment with sophisticated eyes and grasping tentacles. These were the ancestors of modern squid and octopi, already showing signs of the intelligence and behavioral complexity that would make their descendants among the most remarkable creatures in the ocean. The early chordates, our own distant ancestors, were also making their appearance during this remarkable period. These were small, delicate creatures that looked more like primitive fish than anything we would recognize as related to ourselves. But they carried within their simple bodies the basic body plan that would eventually give rise to all vertebrates, a flexible, rod-like support structure called a notochord, a hollow nerve cord running along their backs, and gill slits that could extract oxygen from water. Picaia, one of the earliest known chordates, was only a few inches long and swam through the ancient seas with undulating movements of its entire body. It had no jaws, no paired fins, no complex eyes, just a simple torpedo-shaped body with basic sensory organs at the front end. But this unassuming creature was experimenting with a body plan that would prove incredibly successful, eventually giving rise to fish, amphibians, reptiles, birds, and mammals. The cnidarians, those early masters of tissue organization we met in our previous chapter, were also diversifying rapidly. Some developed into the first large predators, with bell-shaped bodies that could pulse through the water and trailing tentacles armed with sophisticated stinging cells. Others remained attached to the seafloor but developed elaborate tentacle crowns that could capture prey from the surrounding water. Early corals began building the first reef systems, creating complex three-dimensional structures that provided habitat for countless other creatures. These ancient reefs were different from modern ones, built by creatures that are now extinct, but they established the basic ecological principle of ecosystem engineering organisms that create habitat for other species, increasing the overall diversity and complexity of biological communities. Brachiopods developed some of the most beautiful shells of the Cambrian period, with intricate patterns of ridges and folds that increased surface area for filter feeding while maintaining structural strength. Some species developed shells that could open and close like hinged boxes, with complex muscle systems that could create powerful water currents for feeding. The echinoderms, ancestors of modern starfish and sea urchins, were experimenting with radial symmetry and unique water-based hydraulic systems for locomotion. Some early echinoderms had bizarre, asymmetrical body plans that seemed to violate all the rules of animal organization, but they were successful experiments in their time. Even the sponges, those simple animals that seem almost plant-like in their organization, were diversifying into new forms and ecological roles. Some developed sophisticated silica spicules that created beautiful glass-like skeletons. Others experimented with different body forms that could exploit specific types of water flow and food availability. The diversity of body plans during the Cambrian explosion was so great that paleontologists are still discovering new forms and trying to understand their relationships to modern animals. Some creatures seem to combine features from multiple modern groups in ways that challenge our understanding of animal evolution. Others represent entirely novel solutions to biological problems that have no modern equivalents. Among all the innovations of the Cambrian explosion, perhaps none was more profound in its implications than the widespread adoption of bilateral symmetry the moment life discovered left and right, front and back, creating the basic body organization that characterizes the vast majority of complex animals, including ourselves. Before this innovation, most complex animals had radial symmetry, like flowers or wheels, with body parts arranged around a central axis. This organization worked well for sessile creatures that needed to capture food from all directions, or for simple swimmers that moved without a preferred direction. But bilateral symmetry opened up entirely new possibilities for movement, feeding, and interaction with the environment. The advantages of bilateral organization become clear when you consider the challenges of directional movement. A creature with bilateral symmetry can develop a distinct front and back, with sensory organs concentrated at the front to detect food and threats in the direction of travel, and propulsive organs at the back to provide forward thrust. This creates much more efficient swimming and crawling than the undifferentiated movement possible with radial symmetry. Bilateral symmetry also enabled the development of what biologists call cephalization, the concentration of nervous system and sensory organs into a distinct head region. This was revolutionary because it allowed for much more sophisticated information processing and decision-making. Instead of having nerve cells scattered throughout the body, creatures could develop concentrated neural processing centers that could integrate information from multiple sensory systems and coordinate complex behaviors. The head region became the command center of the bilaterally symmetric body, housing not just the brain and major sensory organs, but also the mouth and often the most sophisticated feeding apparatus. This concentration of critical functions at the front of the body made directional movement much more effective. Creatures could literally lead with their most important organs, gathering information and capturing food as they move through their environment. Your own face, symmetrical and balanced, is a love letter to this ancient innovation. The arrangement of your eyes, providing stereoscopic vision that can judge distances with precision, your nose, positioned to sample chemical information from the direction you're facing, your mouth, ready to receive whatever food your forward movement encounters. All of this reflects the same basic body plan that first appeared in the Cambrian seas over 500 million years ago. But bilateral symmetry offered advantages beyond just improved locomotion and feeding. It also enabled much more sophisticated appendage organization. Instead of having appendages arranged randomly around the body, bilateral creatures could develop specialized appendages on different sides and at different positions along the body axis. This allowed for much more complex behaviors and interactions with the environment. The left-right symmetry of bilateral organization also provided developmental advantages. Having two of everything meant built-in redundancy for critical functions. If one eye was damaged, the other could still provide vision. If one side of the nervous system was injured, the other side could potentially compensate. This redundancy made bilateral creatures more robust and better able to survive injury or developmental errors. The evolution of bilateral symmetry required sophisticated developmental control systems. Creating two identical but mirror image sides of the body required precise coordination of gene expression and cellular differentiation. The molecular mechanisms that achieved this coordination were remarkably complex, involving cascades of regulatory genes that could specify left versus right identity and ensure proper symmetry during development. Interestingly, perfect bilateral symmetry is actually quite rare in nature. Most bilaterally symmetric animals show subtle asymmetries that reflect functional specialization. Your own heart, for example, is positioned slightly to the left side of your chest, and your liver is larger on the right side than the left. These asymmetries are carefully controlled and serve important functional purposes, but they represent fine-tuning of the basic bilateral plan rather than departures from it. The success of bilateral symmetry during the Cambrian explosion established it as the dominant form of animal organization. Today, the vast majority of animal species are bilaterally symmetric, from insects to fish to mammals. Even creatures that appear radially symmetric as adults, like sea stars, develop through bilateral larval stages, reflecting their evolutionary heritage. The implications of bilateral symmetry extended far beyond just body organization. It enabled the evolution of complex behaviors, sophisticated sensory systems, advanced locomotion, and ultimately the kinds of neural complexity that would eventually give rise to consciousness and intelligence. In a very real sense, the evolution of bilateral symmetry was a prerequisite for every complex behavior that animals exhibit today. The mathematical elegance of bilateral symmetry has also had profound effects on our human perception of beauty and order. We find symmetric faces and bodies attractive, symmetric designs pleasing, and symmetric arrangements harmonious. This aesthetic preference may reflect deep evolutionary recognition of the importance of symmetry for proper development and function. Sometimes I wonder if that first cell could have imagined. Could she have dreamed of tigers stalking through tall grass? Their powerful bilateral bodies perfectly designed for directional movement and precise strikes. Could she have envisioned hummingbirds hovering at flowers, their rapid wing beats coordinated by sophisticated neural systems that trace their ancestry back to those first simple nerve nets? Could she have foreseen humans lying in bed on quiet evenings, pondering their own beginnings with brains that represent the ultimate elaboration of bilateral organization and cephalization? As you rest there now, breathing softly in the darkness, you are the culmination of nearly 1 billion years of evolutionary elaboration. Every system in your body carries within it the memory of innovations that first appeared during that remarkable flowering of creativity we call the Cambrian explosion. Your eyes, as they grow heavy with sleep, are refined versions of those first sophisticated vision systems that allowed creatures to see their world in detail. The molecular machinery that converts light into neural signals in your retina is essentially the same as that which evolved in the Cambrian seas, though it has been polished and perfected over hundreds of millions of years. Your skeleton, providing structure and protection for your soft tissues, represents the ultimate elaboration of those first experiments in biological architecture. The shells and armor that allowed creatures to grow large and complex while still maintaining their integrity. Your bones are living versions of those ancient protective structures, capable of growth, repair, and adaptation throughout your lifetime. Your limbs, as they relax into comfortable positions, are the descendants of those first jointed appendages that allowed bodies to reach out and manipulate their environment. Every movement you make, from the grossest motor actions to the finest manipulations, depends on the same basic principles of jointed construction that first appeared in the ancient arthropods. Your nervous system, even in sleep, continues the work of information processing and coordination that began with those first simple nerve nets. But where those early systems could only coordinate basic responses to simple stimuli, your brain can create dreams, solve problems, store memories, and contemplate the very evolutionary processes that created it. The bilateral symmetry of your body reflects one of the most successful innovations in the history of life. As you lie there, perhaps on your side, the left-right organization of your organs, the paired symmetry of your limbs, the balanced arrangement of your sensory organs, all of this echoes the body plan that first proved its worth in the Cambrian seas. But perhaps most remarkably, You carry within your genome the same basic toolkit of developmental genes that built those first complex bodies over 500 million years ago. The HOX genes that are organizing your body's repair and maintenance as you sleep are the direct descendants of the molecular architects that first made complex animal life possible. The process of elaboration that began in the Cambrian explosion continues in your own body today. Your immune system learns and adapts, creating new solutions to biological challenges. Your nervous system rewires itself based on experience, literally evolving at the cellular level throughout your lifetime. Your tissues repair and replace themselves, using the same basic mechanisms of cellular cooperation and specialization that first appeared in those ancient seas. You are not just the product of evolution, you are evolution continuing to unfold. Every breath you take represents a successful solution to the problem of gas exchange that was first solved by gill-bearing creatures in Cambrian waters. Every heartbeat reflects the innovation of muscular pumps that first appeared in those early experiments with complex multicellularity. The very consciousness with which you contemplate these ideas represents perhaps the ultimate elaboration of the innovations that began during the Cambrian explosion. Your ability to wonder about your own origins, to feel awe at the complexity and beauty of life, to imagine the experiences of creatures that lived hundreds of millions of years ago, these capacities emerge from the same basic principles of neural organization and information processing that first appeared in those ancient seas. As you drift toward sleep, consider the remarkable journey that connects you to that first cell who chose cooperation over isolation. Through countless generations of elaboration and refinement, through mass extinctions and evolutionary radiations, through changes in climate and geography and chemistry, the thread of life has remained unbroken. Each generation has added its own innovations to the toolkit of life. But the fundamental principles established during those early experiments in multicellularity remain at the core of everything you are. The great elaboration that began in the Cambrian explosion continues in laboratories and hospitals around the world. As humans learn to understand and work with the same biological principles that have been shaping life for hundreds of millions of years, we are beginning to read the genetic code that was first written in those ancient seas, to understand the developmental programs that first built complex bodies, to appreciate the elegant solutions that evolution discovered through patient experimentation over geological time. But even as we unlock the secrets of life through science and technology, the fundamental wonder remains. The improbable journey from single cells to human consciousness, from simple cooperation to the complexity of modern ecosystems, from that first tentative partnership to the rich tapestry of relationships that bind all life together, this remains as mysterious and beautiful as ever. In our next chapter, we will follow this thread of elaboration as it leads from those ancient seas to the very bedroom where you now rest. We will explore how the innovations of the Cambrian explosion were refined and adapted as life moved onto land, developed warm-blooded metabolisms, and eventually gave rise to the consciousness that allows you to contemplate your own remarkable origins. But for now, rest in the knowledge that you are part of an unbroken chain of innovation and creativity that stretches back to the very dawn of complex life on Earth. Every cell in your body carries within it the accumulated wisdom of nearly 1 billion years of evolutionary experimentation. You are not just alive, you are life itself, continuing its ancient journey of discovery and elaboration. The Cambrian explosion was not a single event but an ongoing process, and it continues in you today. As you sleep and dream, your body will repair and maintain itself using the same basic principles that built the first complex animals. Your mind will process and integrate the day's experiences using neural architectures that trace their ancestry to those first simple nervous systems. And tomorrow, when you wake, you will continue the ancient work of being magnificently, elaborately, wonderfully alive. Every creature that has ever lived carries within it the memory of that first moment of togetherness, encoded not just in DNA, but in the very architecture of cellular cooperation that makes complex life possible. As you rest there now, breathing gently in the quiet darkness, you are the living embodiment of an unbroken chain of inheritance that stretches back through deep time to those first tentative partnerships in the ancient seas. The story we've been following together from that single pioneering cell to the magnificent elaboration of the Cambrian explosion might seem like ancient history, disconnected from your immediate experience. But the truth is far more intimate and wonderful. Every system in your body, every breath you take, every heartbeat that pulses through your veins represents a direct continuation of innovations that began nearly 1 billion years ago. The evolutionary relationships that connect all animals are like an enormous family tree, with countless branches spreading outward from common ancestors, but all ultimately traceable back to those first multicellular partnerships. When scientists study the DNA of different animals, they can read the story of these relationships written in the genetic code itself, seeing how slight variations accumulated over millions of years to create the incredible diversity of animal life we see today. Your own genetic code carries within it sequences that are virtually identical to those found in creatures as different as sea anemones and fruit flies, testament to the shared ancestry that unites all complex life. The genes that control your basic body plan development, those HOX genes we explored in our last chapter, are so similar to the equivalent genes in other animals that they can sometimes be swapped between species with little effect on development. But the connections go deeper than just shared genes. The fundamental principles of cellular organization that we discovered in our earliest chapters, the ways cells adhere to each other, communicate through chemical signals, and coordinate their activities for the benefit of the whole. These same principles are operating in your body right now, refined and elaborated, but essentially unchanged from their ancient origins. The calcium-binding proteins that allow your muscle cells to contract in coordinated waves are molecular cousins of the proteins that first enabled those early colonial organisms to move together through primordial waters. The ion channels that carry electrical signals through your nervous system are descendants of the first cellular communication systems that allowed simple multicellular creatures to coordinate their responses to environmental changes. Even the basic rhythm of your metabolism, the cycles of energy production and consumption that keep your cells functioning, echoes patterns that were established in those first experiments with multicellular cooperation. Your cells still divide using essentially the same mechanisms that allowed that original pioneer to create the first lasting partnership, though the process is now embedded within the complex regulatory systems that ensure proper development and tissue maintenance. The beauty of evolutionary relationships lies not just in their antiquity, but in their continuity. You are not merely descended from those ancient creatures, you are, in a very real sense, their direct continuation. The same fundamental processes that kept those first partnerships alive and growing are still operating within you, adjusted and refined by hundreds of millions of years of evolutionary fine-tuning, but never fundamentally altered. At some point in this grand story, our particular lineage made another bold choice: to build a backbone. This decision, made by small, swimming creatures in ancient seas over 500 million years ago, would prove to be one of the most successful architectural innovations in the history of animal life, eventually giving rise to fish and frogs, dinosaurs and eagles, whales and humans. The earliest vertebrates were humble creatures, not much to look at by modern standards. Picture something like a small, translucent eel swimming through Cambrian waters. Its body supported not by a true backbone, but by a flexible rod called a notochord. This simple structure was revolutionary because it provided a stable platform for muscle attachment while still allowing the fluid, undulating movements that made efficient swimming possible. These early chordates, as scientists call them, were experimenting with a fundamentally different approach to body support than the external skeletons favored by arthropods or the hydraulic systems used by many other creatures. Instead of wearing their structure on the outside or relying on internal pressure, they developed an internal framework that could grow with the animal and provide support without limiting flexibility. The transition from these early chordates to true vertebrates, animals with actual backbones made of individual vertebrae, was gradual and beautiful. Over millions of years, the simple notochord became reinforced with cartilage, then bone, creating a jointed internal skeleton that combined strength with remarkable flexibility. Each vertebra could move slightly relative to its neighbors, allowing the backbone to bend and twist while maintaining structural integrity. But the backbone was more than just a support structure. It also protected the spinal cord, that crucial extension of the brain that carried signals throughout the body. This protection allowed for the evolution of increasingly sophisticated nervous systems, as delicate neural tissue could develop without fear of damage from the mechanical stresses of movement. The first fish, those pioneering vertebrates that truly mastered aquatic life, developed this basic body plan into forms of exquisite efficiency and beauty. They streamlined their bodies for minimal water resistance, developed sophisticated fin systems for precise maneuvering and created sensory systems that could detect the slightest vibrations and chemical traces in the water around them. Some early fish developed jaws, transforming from passive filter feeders into active predators capable of biting and tearing their prey. This innovation opened up entirely new ecological niches and drove rapid diversification as different lineages specialized for different types of prey and feeding strategies. The transition from water to land represents one of evolution's most remarkable achievements. Around 370 million years ago, some fish began to venture into shallow waters and eventually onto the shores of ancient continents. These pioneering amphibians faced enormous challenges, supporting their body weight against gravity without the buoyancy of water, breathing air instead of extracting oxygen from water, preventing their skin from drying out in the harsh terrestrial environment. The solutions they evolved were ingenious. Fins became limbs with jointed digits that could grip uneven surfaces and support body weight. Gills were supplemented and eventually replaced by lungs that could extract oxygen from air. Skin developed protective layers and glands that could maintain proper moisture levels. The vertebrate body plan proved remarkably adaptable to this entirely new way of living. Reptiles took the terrestrial adaptation even further, developing scaly, waterproof skin and eggs that could develop on dry land, freeing them from the need to return to water for reproduction. Some reptiles grew to enormous sizes, becoming the dinosaurs that dominated terrestrial ecosystems for over 160 million years. From these reptilian ancestors arose two remarkable innovations that would prove crucial for the development of more complex behaviors and eventually consciousness itself: warm-bloodedness and advanced parental care. The first mammals were small, mouse-like creatures that developed the ability to regulate their own body temperature and nurture their young with milk produced by specialized glands. Each of these transitions from water to land, from cold-blooded to warm-blooded, from egg-laying to live birth and milk production, represented not just changes in lifestyle, but fundamental reorganizations of the vertebrate body plan, all built upon that original innovation of internal skeletal support that first appeared in the ancient seas. And eventually, our story leads to creatures with warm blood and beating hearts, hearts that pump with the rhythm first established by those ancient cellular partnerships we encountered at the very beginning of our journey together. The mammalian heart is one of evolution's most remarkable achievements, a four-chambered pump of extraordinary efficiency and endurance that beats roughly 3 billion times over the course of a human lifetime. But its fundamental operating principle, coordinated cellular contraction driven by electrical signals, traces directly back to those first simple muscle cells that appeared in early multicellular organisms. Your heart, beating steadily as you rest there in the quiet darkness, is essentially a sophisticated version of the cellular cooperation that we've been exploring throughout our journey. Each cardiac muscle cell maintains its own intrinsic rhythm, but they all synchronize their contractions through gap junctions, those same cellular communication channels that first allowed simple colonial organisms to coordinate their activities. The electrical system that coordinates your heartbeat is a marvel of biological engineering. Specialized pacemaker cells generate rhythmic electrical impulses that spread through the heart muscle in precisely timed waves, ensuring that the chambers contract in the proper sequence to maximize pumping efficiency. This system is so reliable that it continues to function even when disconnected from the brain, carrying within each cell the memory of the rhythm that has sustained vertebrate life for hundreds of millions of years. The four-chambered structure of the mammalian heart, with separate chambers for receiving and pumping both oxygen-rich and oxygen-poor blood, represents the culmination of a long evolutionary process that began with simple, contractile vessels in early animals. Fish hearts have two chambers. Amphibian and reptile hearts typically have three, and mammals and birds independently evolved four-chambered hearts that completely separate the pulmonary and systemic circulations. This separation is crucial for maintaining the high metabolic rates that make warm-bloodedness possible. Your heart can generate the pressure needed to push blood through your lungs to pick up oxygen, then through your entire body to deliver that oxygen to tissues that are constantly consuming it at high rates to maintain your body temperature and power your active lifestyle. The mammalian innovation of warm-bloodedness, the ability to maintain a constant, elevated body temperature regardless of environmental conditions was revolutionary because it freed animals from dependence on external heat sources. This metabolic independence allowed mammals to be active at night, in cold climates, and during seasonal changes that would immobilize cold-blooded creatures. But warm-bloodedness came at a cost. It required a much more sophisticated circulatory system to deliver the nutrients and oxygen needed to fuel constant, high-rate metabolism. Your heart works much harder than that of a similarly sized reptile, pumping blood through a more extensive network of vessels to support tissues that consume energy at rates that would be impossible for cold-blooded animals to sustain. The regulation of your heartbeat reflects the integration of multiple systems that first appeared at different points in vertebrate evolution. Your autonomic nervous system can speed up or slow down your heart rate based on your activity level and emotional state. Hormones like adrenaline can prepare your cardiovascular system for intense activity. Even the chemistry of your blood, its pH, oxygen content, and electrolyte balance influences the strength and rhythm of your heart's contractions. Which brings us, finally, to you lying there, breathing softly, your body a living testament to nearly 1 billion years of cellular cooperation, innovation, and refinement. You are not just the descendant of all those ancient creatures we've encountered in our journey. You are their direct continuation, carrying within your very cells the accumulated wisdom of evolutionary experimentation across geological time. The story of human evolution is often told as a series of dramatic transitions from ape to human, from quadruped to biped, from simple tool use to complex technology. But the deeper truth is that these recent changes, remarkable as they are, represent only the latest chapter in the much longer story we've been following together. Your basic body plan, your cellular organization, your fundamental life processes, all of these connect you directly to creatures that lived hundreds of millions of years ago. How human development recapitulates ancient patterns is one of the most beautiful demonstrations of evolutionary continuity. In your mother's womb, you briefly resembled those ancient ancestors, as if life were telling its whole story again, just for you. During the earliest stages of your development, you were essentially a hollow ball of cells, not unlike those first simple multicellular organisms that floated in Cambrian seas. As your embryonic development progressed, you temporarily developed gill slits, even though you would never need to breathe water. You had a tail, though it would be reabsorbed before birth. Your limbs began as simple buds that gradually developed the jointed structure pioneered by those first vertebrates that ventured onto land. Your nervous system formed from the same basic pattern, a hollow tube along your back that characterized the earliest chordates. This developmental recapitulation is not mere coincidence. It reflects the fundamental conservatism of the genetic toolkit that builds complex bodies. The same regulatory genes that controlled development in ancient animals still control your development today, activated in the same basic sequence, creating the same fundamental patterns, though elaborated and refined in countless ways. Your brain, that remarkable organ that allows you to contemplate your own origins, represents perhaps the ultimate elaboration of the nervous system innovations that first appeared in simple animals like jellyfish and early worms. The basic principle, electrical signaling between specialized cells, remains the same, but the complexity and sophistication have been amplified beyond anything that existed in those ancient seas. The human brain contains roughly 100 billion neurons, each connected to thousands of others, creating a network of unimaginable complexity that emerges from the same basic cellular cooperation principles we've been exploring. When you form a memory, learn something new, or have a creative insight, you're experiencing the culmination of nearly 1 billion years of evolutionary experimentation with cellular communication and coordination. Your consciousness itself, your ability to experience, to wonder, to feel awe at the beauty of the natural world, represents something entirely new in the history of life on Earth. Yet it emerges from the same fundamental processes of cellular cooperation that began with those first tentative partnerships in ancient seas. The neurons firing in your brain as you listen to these words are using the same basic mechanisms of electrical signaling and chemical communication that first appeared in primitive nerve nets hundreds of millions of years ago. Even your capacity for language, for abstract thought, for imagining the experiences of creatures that lived long before you were born, builds upon much more ancient foundations. The social cooperation that makes human culture possible reflects the same basic principles that first made multicellular life successful: specialization, communication, coordination for mutual benefit. And the story isn't over. Even now, in your body, cells are dividing, specializing, cooperating in ways that would make those ancient pioneers proud. Your immune system learning, your brain rewiring itself with new thoughts and dreams, your tissues repairing and maintaining themselves. All of this represents the ongoing continuation of the evolutionary processes that began nearly 1 billion years ago. As you sleep tonight, millions of your cells will divide to replace those that have reached the end of their lives. Each division will use essentially the same mechanisms that allowed that first cell to create lasting partnerships with her daughters. The molecular machinery that copies your DNA, separates the copies, and divides your cells into two identical daughters is directly descended from the systems that made multicellular life possible in the first place. Your immune system is constantly learning and adapting, creating new solutions to biological challenges in real time. When your immune cells encounter new threats, they undergo rapid evolution within your body, generating countless variations and selecting the most effective responses. This process of variation and selection mirrors the larger evolutionary processes that shaped life over geological time, but compressed into days or weeks rather than millions of years. Your brain continues to reorganize itself throughout your life, forming new connections between neurons, strengthening pathways that are frequently used, and pruning those that are no longer needed. This neural plasticity allows you to learn new skills, form new memories, and adapt to changing circumstances. It represents a kind of ongoing evolution at the cellular level, with the same basic principles of adaptation and optimization that have driven life's development since the very beginning. Even your dreams, as you drift towards sleep, represent a continuation of ancient processes. The neural activity that creates dream experiences uses the same basic mechanisms of electrical signaling and pattern formation that first allowed simple nervous systems to process information and coordinate responses. Your dreaming brain is essentially playing with the same tools that evolution has been using for hundreds of millions of years to solve the challenges of complex life. The cells lining your digestive system replace themselves every few days, using stem cell systems that maintain the same basic architecture while constantly renewing the cellular workforce. Your skin regenerates continuously, with new cells migrating from deeper layers to replace those lost at the surface. Your blood cells are manufactured in your bone marrow at a rate of millions per second, each one specialized for its particular function but all working together to maintain the integrity of your internal environment. All of these ongoing processes in your body represent the continuation of the story we've been following together. The same principles of cellular cooperation that first appeared in those ancient seas continue to operate within you, refined and elaborated, but fundamentally unchanged. You are not just the product of evolution, you are evolution continuing to unfold, adaptation and innovation happening in real time within the community of cells that is your body. The story that began with a single cell choosing cooperation over isolation continues in every relationship you form, every act of kindness you perform, every moment when you choose to work together with others rather than pursuing purely individual goals. The same principles that made complex life possible, specialization, communication, coordination for mutual benefit, continue to shape human societies and cultures. As you rest there now, breathing softly in the darkness, you carry within you the memory of that first moment of togetherness, encoded not just in your genes but in the very structure of your being. You are the universe becoming conscious of itself, life reflecting on its own remarkable journey, from simplicity to complexity, from isolation to community, from that first tentative partnership to the rich tapestry of relationships and experiences that make up human existence. The long journey home from ancient seas to your bedside is complete, but it's also just beginning. Every day you live adds new chapters to this ancient story, new innovations in the ongoing experiment of conscious, cooperative life. You are both the inheritor of nearly 1 billion years of evolutionary wisdom and the creator of new possibilities for what life can become. Sleep well, knowing that you are part of something magnificent and ongoing, something that began in the depths of ancient seas and continues in the beating of your heart, the firing of your neurons, the cooperation of your trillions of cells as they work together to sustain the miracle of your conscious existence. So when we asked, how did a single cell build the entire animal kingdom? What we were really asking was, how did cooperation become the secret of life? The answer is beautifully simple. By choosing to stay together, by learning to work together, by specializing and supporting each other. That first cell's gift wasn't just her genes, it was the idea that we could be stronger than I. Right now, as you listen to these words, trillions of cells are cooperating to keep you alive, to let you think, to help you dream. You are not just made of stardust, you are made of cooperation itself. And perhaps the most beautiful part is that we're still becoming, still evolving, still learning new ways to work together. Not just as cells in a body, but as humans sharing a planet, sharing stories, sharing quiet moments like this one. That single cell, floating in an ancient ocean, made a choice that would echo through deep time. She chose connection over isolation, cooperation over competition. And now, nearly 1 billion years later, you carry that choice forward every day. So as you drift towards sleep tonight, maybe take a moment to thank that ancient ancestor. Thank her for choosing to stay together, for beginning this impossible, beautiful story that led, eventually, to you. In the beginning there was one. Now there are billions, each carrying the memory of that first moment of togetherness in every cell, every heartbeat, every dream. Sleep well, fellow travelers. The story continues tomorrow. Until next time, keep wondering, keep marveling, and remember, you are living proof that cooperation is the most powerful force in the universe.